Well, I am happy you've all joined us today. Immunology is a huge topic. There is far too much for us to cover in just 1 hour. Um, but it is fascinating and it's key to our understanding of HLA. And so I hope you'll come along with me as we go through a very brief overview of certain aspects that are important in our understanding of immunity and of the science of HLA. So, we will start with a brief overview of the immune system. We will talk about immune communication and cascades. We'll talk about immune organs and tissues, immune cells, and the role of HLA in immunity. First, beginning briefly with the immune system. We have certain aspects that are innate. They are physical barriers, phagocytosis, which is cells which can ingest pathogens, cytokines, which are chemical messengers, protein messengers that stimulate further actions by the immune system, and other mechanisms. So, innate immunity is the first line of defense for the body. It can trigger inflammation, cell killing, and then activate the adaptive immune response. There are certain patterns that are recognizable as foreign, even without the specificity of adaptive immunity, and that's where innate immunity comes in. Um, Past that point, we can develop adaptive immunity, which includes specificity and memory. It is specific to a particular pathogen or microbe or foreign antigen. Um, and we are capable of mounting an initial primary immune response. And if we see that same specific antigen again, we can have a secondary, much stronger immune response. Now, we'll go into much more detail on this as we go through our slides, but here's an example of how adaptive immunity might be sparked. So, in the case of a virus infecting our cells, a particular type of immune cell can ingest that microbe and then migrate into an area of the lymph node where we have concentrated immune cells, where there can be communication among them. They can activate further cells, T cells that can go towards killing virus infected cells, B cells, which can stimulate antibody production and create antibodies, which can neutralize viral antigens. So we'll go into greater detail as we go, but that's a bit of a summary where there's multiple cellular interactions to create a specific response that can retain memory. When we're looking at immunity, the goal is to have a balance. Too much of an immune response can lead to hypersensitivity reactions and autoimmune diseases. Now, there are multiple types of hypersensitivity reactions, some of which are what we would think of as allergies or anaphylactic responses, some of which are mediated by antibodies binding to cells, which is important in transfusion and transplantation reactions. There are immune complex mediated reactions similar to lupus, and T cell mediated reactions similar to Stevens-Johnson syndrome. So, these are some of the ways that too much immune reactivity can actually cause harm to our cells on the side. There are also endless numbers of autoimmune diseases, and these are only some, but when we unfortunately have immune responses against our own tissues, we can have some uh, disease responses, some of which are also associated with HLA. So, too much of an immune response is a problem. Too little of an immune response can also be an issue. Of course, if we can't respond to a germ or a pathogen, we will get sick, we'll have illness, but we also can suffer from immune deficiencies or cancer if we have too little immune response. There are multiple types of immune deficiencies that may be something we're born with due to a genetic issue. And there also may be secondary immune deficiencies that occur because of infections, drugs, or malnutrition, etc. Additionally, our immune cells help fight off and kill precancer cells. And if we become immune suppressed, we may lose some of that capability and we're more likely to deal with a cancer situation. So, the right balance of immune activation and inhibition keeps us healthy. Right. Moving now to immune communication and cascades, we'll talk about how our body knows to proceed with some of these processes. So, immune regulation is reliant on protein messengers called cytokines. Cytokines are produced by multiple types of cells and may have an activating or inhibitory effect on immune responses. Important cytokine groups include interleukins, colony stimulating factors, interferons, tumor necrosis factors, transforming growth factors, and chemokines like CXCL and others. These cytokines can have uh, effects on the immediate cells around them, cells at some distance, or cells and systems at a great distance from them. So, the inflammation cascade is an example of cytokines in action and is part of our innate immunity. Inflammation causes tissues to become red, warm, painful, and swollen as blood and immune cells are recruited to the site of an infection or wound. White blood cells can ingest the invaders and release pro inflammatory cytokines like interleukin 1, interleukin 6, and TNF alpha. These cytokines are signaling molecules. They attract and activate other immune cells to the site of inflammation. These cells join in additional cellular and cytokine responses, extending the cascade. The activated immune cells ingest and destroy pathogens or cellular debris and release toxic molecules to kill the pathogens. Once the threat is eliminated, anti inflammatory cytokines such as IL-10 and TGF beta are released to reduce the inflammatory response and promote tissue healing and repair. Excessive or prolonged inflammation can cause tissue damage and contribute to the development of chronic inflammatory diseases. Certain immune cascades require multiple signals from membrane bound receptors to start a response. Lymphocytes are a specialized type of white blood cell that include T cells, which we'll talk about extensively in this session. They require the binding of multiple membrane receptors to be activated. So, we'll discuss this, but T cell receptors require interaction with a peptide bound to an MHC molecule. Additionally, CD4 or CD8 molecule binds to stabilize the interaction, and there is an interaction between molecules called a CD28 and a B7, not to be confused with an HLA B7, but there's a marker called B7 that also sometimes is called CD80. But um, all three of those signals are required to uh, for the cascade of further action to happen, whether to attack a pathogen or to tolerate an antigen that is present. If activated, if that T cell sees a foreign peptide presented with all of these co-stimulatory signals, that T cell can become activated and initiate cell killing and cytokine release, which stimulates B cells to produce antigen-specific proteins called immunoglobulins or antibodies. The antibodies can bind and neutralize an antigen. They can flag cells to be ingested and destroyed, and they can activate additional protein cascades to kill cells. One of those ways that the cells can be killed is through the complement system. The complement cascade was named because it complemented the immune processes of the cells. It is part of both the innate and adaptive immune responses. These pathways can amplify their own production. They can recruit immune cells. They can form complexes that burst cell membranes. The final grouping of proteins is called the membrane attack complex, and it can pierce pores in a cell and burst it. But each of these, the classical pathway, the lectin pathway, and the alternative pathway, can cycle into each other. It involves the cleaving and binding of various proteins in a continuous cascade to have these further effects. The classical pathway of the complement system is part of the adaptive immune system. It's reliant on antigen specific antibodies, which bind to the cell and then trigger a series of proteins to cleave and bind and carry on with this signaling cascade. The result of this is cell killing mediated by the C1q complement molecule bound to antibodies on the cell surface. So I will just highlight, if you can see in our image at the top, we have the invading pathogen, we have the antigen on the cell, we have the antibodies bound to it, and then we have our C1q molecule, um, which is which is bound, and from there, the cascade will continue until the cell can be killed. This complement system has been quite important in the history of HLA testing. We have a session coming up on complement dependent cytotoxicity testing and other historical assays. But I do want to mention that this process has been part of the CDC testing, where um, mixing cells in serum with complement and fluorescent dye could define antibody. It could type for particular HLA types, and is quite key um, in the history of transplantation. Additionally, important aspects of the complement system in transplantation, for many years, C4D deposition has been a marker on biopsy of rejection. And so often C4D staining will be done on biopsy sections. Now we are we have available to us C1q antibody testing for antibodies which will fix complement. And we also have new assays coming up that are able to find rejection before you have so much damage and so much cell killing. But the source of this comes from those processes where you have antibodies bound to an antigen on the cell, and then you have this continual cascade of these proteins cleaving and binding until the trigger of the cell being is the the killing of the cell is triggered. Excuse me. Moving on now to immune organs and tissues. First, the skin is an essential element of the immune system. It's a barrier against infection. There are also various mucus secretions and cilia, and various ways that we can be protected from germs of various types entering. We also have immune organs, including the bone marrow, where red and white blood cells originate. The B cells also undergo most of their development in the bone marrow. The thymus, which is a gland behind the sternum in the chest, which is where the T cells mature. Lymph vessels, which filter and drain fluid from cells and tissues into the lymph nodes. This is in conjunction, we have our circulatory system. We also have our lymph circulatory system. Lymph nodes are centers which contain large numbers of immune cells and provide a site to concentrate and attack pathogens. We have mucosal associated lymphoid tissue or MALT located in the tonsils, the intestine, the appendix, and other areas. These contain lymphoid follicles, which can also bring pathogens in contact with concentrated immune cells. The spleen stores healthy red blood and white blood cells, um, filters out dying cells and pathogens, and serves as a location for immune cell interactions and activation. Now, moving on to those immune cells. Cells of the blood and the immune system originate in the bone marrow as hematopoietic stem cells, or HSCs. Multipotent HSCs can differentiate into myeloid or lymphoid cell lines. Now, an important key is we often identify cells by clusters of differentiation or CDs. These are markers on the cell that we can probe for to identify which cell we're dealing with. So, multipotent HSCs can be identified by the presence of a cell surface marker that we call CD34. Mature white blood cells that are no longer that are already differentiated into whatever they're going to end up as. They have progressed past the stem cell phase. They express CD45 and no longer express CD34. So, one of the cell lines is the myeloid cell line that can descend from that. Myeloid cells include erythrocytes or red blood cells, which carry oxygen through the body. They have many antigens that are important in transfusion medicine. Um, Myeloid cells also include megakaryocytes, which produce platelets. Um, those platelets clot in the blood in response to injury and also have their own system of antigens. A subset of myeloid cells are called granulocytes. They contain small vesicles of enzymes that are released during immune responses. You'll sometimes hear these referred to as polymorphonucleocytes or PMNs because they have uh, multi-lobed nuclei that can appear in different ways. Neutrophils are the most common type of white blood cells. They activate quickly during inflammation. They're phagocytic, so they can engulf the pathogens they encounter as well as release toxic granules. Eosinophils have granules with toxic peroxidase. They are involved in fighting parasitic infections, allergies, and asthma. Basophils are involved in allergic responses and asthma with granules containing histamine and heparin. The names of the neutrophils, eosinophils, and basophils come from the dyes that they stained with under a microscope when first being discovered. Further myelocytes include the monocytes, which differentiate into dendritic cells and macrophages. All of these cells are phagocytic, so they can engulf and destroy pathogens. They also process and present peptides to the immune system to verify whether the peptides should be tolerated or attacked. So we're done with the myeloid cell line. Now we move on to the lymphoid cell line. The lymphocytes, more unique immune cells. So another specialized group of white blood cells. Among them are natural killer cells or NK cells, which defend against viruses and cancer cells and can respond to threats without previous exposure to the antigen. So they can notice uh, something that doesn't belong there and attack it without having a specific receptor for it. In addition to toxic granules, they can release cytokines to recruit more immune cells, and they may be involved in activating or suppressing immune responses depending on the signals that they get. NK cells can be identified by the presence of CD16 and 56 on the membrane. Moving on now to T cells. T cells are specialized lymphocytes that express CD3 and specific T cell receptors or TCRs. These T cell receptors are specific to an antigen, um, and we'll talk more about that coming up. T cells differentiate into additional types. Cytotoxic T cells, which express CD8, they can directly kill diseased cells. CD4 helper T cells, which recruit macrophages. They recruit CD8 T cells and B cells, which can then begin antibody production. Regulatory T cells, which can reduce the immune response to protect us from damaging our own cells. Memory T cells, which can be reactivated to attack the same antigen if it appears again later. So, if we do see uh, the same antigen again, those memory T cells can reactivate into cytotoxic T cells, um, and the cycle begins again. So, as T cells develop, the immature T cells migrate from the bone marrow to the thymus, where they undergo rearrangement of the TCR alpha and beta genes to increase diversity and specificity. They then go through a selection process where they have to be able to recognize self antigens presented by cells within the thymus. The T cells that can recognize self antigens as something to tolerate, um, if they can recognize those self antigens with the MHC or HLA complex, they are allowed to grow and continue. Any cells that can't engage with self antigens and MHC go through apoptosis and are eliminated. Next, there is a negative selection where if they've passed the first stage, they will be exposed to a variety of self antigens presented by antigen presenting cells. If they strongly react and attack those self antigens, they are eliminated to prevent autoimmunity. So we're looking for the sweet spot of not too little and not too much to make a T cell with a specific receptor that reacts the correct amount. The cells then undergo differentiation. After passing through those selection series, the T cells differentiate into subset depending on the type of T cell receptor they express, and they'll begin expressing a CD4 or CD8 or the appropriate additional markers. And then they migrate out of the thymus and enter the bloodstream where they circulate through the body to carry out their immune functions. Now, additional lymphocytes are the B cells. They express CD19 and CD20 and specific B cell receptors. They also differentiate into additional types plasma cells, which produce and secrete antibodies, memory B cells, which can be reactivated to attack the same antigen if it appears again later. B cells mature in the bone marrow and they undergo a similar gene rearrangement as T cells to create a diverse repertoire of B cell receptors. So there is a rearrangement and assembly of immunoglobulin gene segments, which encode the variable regions of BCRs responsible for antigen recognition. Now, a B cell receptor is also an antibody, but it's membrane bound. And so until the B cell is activated to turn into a plasma cell and start secreting antibodies of the same specificity, the BCR acts as the initial antibody. So, um, there are various segments in the germline DNA. There's a variable diversity and joining segments. They can be arranged in different orders. So, this is yet another way that our immune system adds variability in the types of pathogens we can fight, in the types of foreign molecules we can bind with and respond to. And so, you get significant variability, significant possibilities in this somatic recombination as your body is creating these B cell receptors. So, if they successfully generate functional BCRs, they undergo a similar selection process to ensure self tolerance and avoid strong autoimmunity. Mature B cells migrate from the bone marrow to secondary lymphoid organs, such as the spleen and the lymph nodes. In these organs, the B cells encounter antigens, and if their BCRs recognize the antigen, they become activated. Or if they are stimulated by cytokine signals and other immune processes. So, activated B cells become plasma cells, which produce large amounts of antibodies specific to the encountered antigen. Some activated B cells also undergo class switching and affinity maturation to produce more potent and specific antibodies. And then the plasma cells secrete those antibodies into the bloodstream where they can bind to pathogens and neutralize them, mark them for destruction by other immune cells, or activate complement. Now, looking at a general antibody structure, uh, there are two light chains and two heavy chains. They are held together with disulfide bonds. Um, it is shaped like the letter Y in the English language, but what you'll see is at the top of there are the antigen binding sites, sometimes called complementarity determining regions or hypervariable regions, that have these unique places that are is where they engage with the antigen. So we have the antigen binding sites, and we sometimes refer to this region as the FAB region or FAB region, which refers to fragment antigen binding. There's a hinge to allow some amount of flexibility. And then the constant region we call the FC region. Originally, that meant fragment crystallizable when it was being discovered. Now you can think of it as being fragment complement binding, if you'd like. So, antibodies bind to specific regions of antigens called epitopes. You can imagine whether this is a protein on the outside of a cell or a protein, a soluble protein in the blood, that the antibody isn't actually binding to the whole entire antigen. It's binding to a particular section, and that may be partly because of the way the protein is folded, a conformational epitope, but there is a specific region where the binding capacity of the CDR or the antigen binding fragment of the antibody is complementary to the binding capacity of the, you know, i- uh, amino acid sequence, the folding, the binding capacity, the side chain properties, etc. And so where you have these interactions, you have the antibody binding to an epitope that is a specific region of an antigen that it is reactive to. So we have the epitope, which is part of the antigen that can interact with the molecule, with the antibody molecule. The part of the antibody that interacts with the epitope is called the paratope. There are three binding regions on the heavy chain and three binding regions on the light chain. So there are six total spots on the paratope. Sometimes we think about the idea of affinity of an antibody or avidity of the antibody. Affinity is the strength of the binding between one epitope and one paratope. There might be multiple bonds, there might just be one, depending on the chemical composition. But avidity is the combined strength of all of the binding sites on a single antibody. So you may have a stronger interactions overall if you have multiple paratopes involved. Um, Antibodies may undergo a process called somatic hypermutation, which increases their avidity and makes them bind more tightly when produced by the plasma cells. Antibodies come in different varieties that we call isotypes. IgM is the first antibody produced during an immune response. It is present in the blood and helps activate the complement system and recruit other immune cells to eliminate pathogens. IgD is present in low levels in the blood, but is expressed on the surface of B cells. It is believed to play a role in B cell activation. IgG is the most abundant antibody in the blood and is the main antibody involved in secondary immune responses. It can cross the placenta and help provide passive immunity to the fetus. Um, it is the the antibody that interacts most because it's in the highest concentration. Um, IgA is primarily found in mucosal secretions such as saliva, tears, and breast milk. Again, it can pass from mother to child to help protect a baby before they've developed their own immunity. And IgE is involved in allergic reaction and defense against parasites. It binds to allergens and triggers the release of inflammatory molecules, leading to allergic symptoms. And prior to that, it was originally discovered in the context of fighting off helminthic worm infections. So if you have allergies, maybe we just need to be grateful that we don't have parasites. IgM, IgA, and IgG can bind complement. IgM is the most effective at binding complement. But what will happen in the course of an infection is that IgM is the first antibody that is created, and then again, there's a process that we refer to as class switching, where initially a lot of IgM will be created, and then the plasma cell will switch to producing more IgG. Additionally, a few of the isotypes have some unique features. IgM occurs in a pentamer with 5 subunits. These are very effective at activating complement because of their larger size. It only takes one IgM molecule to activate complement. IgE appears as a dimer with 2 subunits. And IgG has specific subclasses with variable characteristics, including variable hinge region flexibility that can influence their efficiency at binding complement. IgG1 and IgG3 are the most effective at binding complement. And for those who are interested in antibody testing or reasons where we might be interested in cleaving the connections between the IgM molecule with something like DTT. Um, the pentamer is held together with disulfide bonds, just like the IgG molecules, but following a procedure with validated concentration and incubation time means that you can successfully cut apart an IgM molecule without also cutting apart your IgG molecules. So, moving on to the role of HLA in immunity. HLA stands for human leukocyte antigens. It's also called the major histocompatibility complex, or MHC. They are proteins that present peptides to the immune system. They induce and regulate immune responses and help distinguish between self and non self. So, if you joined us for our last webinar, we have the surname family here, father and mother, who've passed on their chromosome 6 to a couple of children. Now, we think of haplotype inheritance because each individual has 2 chromosome 6s. When we pass on genes to our children, we pass on only half of our genes. So a child receives half of their chromosomal content from their mother and half from their father. So uh, when we think about that chromosome 6 inheritance, we have a lot of information included in that. So that is the location of the immune genes of the major histocompatibility complex. Class 1 MHC includes HLA A, B, C, E, F, and G. Class 2 MHC includes HLA DR, DQ, DP, DN, DM, and DO. And HLA class 3 includes the complement proteins C2 and C4 and genes for tumor necrosis factors and heat shock protein 70, which are cytokines. Classical MHC includes HLA A, B, C, DR, DQ, and DP. And non-classical MHC genes have additional roles in the immune system, but they'll not be covered here. So, HLA class 1 has 3 alpha chains and a constant beta 2 microglobulin. It constitutes HLA A, B, and C, and it can bind shorter peptides in the binding groove. And when those are present, they will be somewhat flexible but anchored by particular bound points within that groove. HLA class 1 is expressed on all nucleated cells and platelets. So human leukocyte antigens doesn't really describe these proteins as they're present on almost all the cells of the body. The HLA class 1 pathway is that HLA class 1 presents intracellular peptides to CD8 positive cytotoxic T cells. This is most common for inherent self proteins or viral proteins that have infected the cell. So the antigen will be present in the cytosol. The antigen would be processed and chopped up and then transferred to the endoplasmic reticulum where the MHC molecule is being assembled. So the peptide will be integrated into the molecule. It's part, it's contained within it. It would be denatured if it was pulled apart. So it will go out in a vesicle and then be expressed on the cell surface. So if that is a self antigen, it will be expressed and tolerated. If that is a foreign antigen, a viral antigen, it will bind with that T cell receptor, and that will start a cascade to initiate cell killing, initiate cytokine production, and additional immune processes. Something else to remember about HLA class 1 is that HLA class 1 is also one of the receptors for NK cells, receptors that are called killer immunoglobulin like receptors or KIR. So they're KIR receptors that are somewhat like antibodies, but not all the way. So NK cells have a series of genes, some of which are or are inhibitory and some of which are stimulatory to the immune system. They have a series of ligands or receptors that they have to bind to, and they will take their signal from the combination of those signals. So NK cells are inhibited by healthy expression of self antigens and are activated in the case of missing or upregulated self HLA. So, in cases where maybe a cancer cell has reduced or eliminated expression of HLA, they would notice that and try to attack it. Maybe if a cell is expressing foreign HLA but not self HLA, it might attack it. Um, or maybe if a cell is inflamed and is expressing too much of its own HLA. But that is a way that NK cells can be activated without having the specificity of receptors that happen with T cell receptors and B cell receptors. So, moving on to HLA class 2, we have 2 alpha chains and 2 beta chains. Again, you have the peptide binding, but you can bind slightly longer peptides in a class 2 molecule, and you have these are the DR, DQ, and DP molecules. These are expressed only on antigen presenting cells. So these are on B cells, these are on dendritic cells, macrophages. Langerhans cells are tissue specific, gut specific, digestive specific macrophages. There are some additional tissue specific macrophages, but the idea about these is that they can ingest the pathogen. So they are more likely to be associated with bacterial infections as they've ingested the germ. So HLA class 2 presents extracellular peptides to CD4 positive helper T cells. Again, self antigens presented in this way are tolerated. So an extracellular protein will be taken up. It will be broken up into peptides. The MHC class 2 with a blocking molecule basically will leave the endoplasmic reticulum in a vesicle. Those two vesicles will bind together and the peptide be combined into the class 2 molecule. That'll move up and be expressed on the cell with the appropriate cofactors, and then will either be tolerated or that foreign antigen will stimulate further inflammation, will stimulate cell killing, and the production of antibodies. So, what we talk about when we talk about HLA, we might talk about what protein is on the outside of the cell. We might talk about what genes created that protein. We might talk about the sequence of the peptide binding groove because we may be interested in what peptides can be bound and presented to the immune system. We have the idea of an immune repertoire. What are the germs that someone can fight? What are the antigens that they can present? And so we may be interested in the sequence at the peptide binding groove. We may be interested in the specific sequences where the T cell receptor contacts the molecule, where we have a little bit of variability, and you have that receptor interaction. We might be interested in where a B cell receptor or an antibody could bind and potentially um, inflame the body, cause complement fixation, etc. So when we're talking about HLA, we may be interested in multiple of these aspects, but the key piece is that to stimulate the adaptive immune response, you need to have those molecules expressing the antigen or, displaying the peptide to the immune system to check if it should be tolerated or if it should be fought. So, um, our multi-layered immune systems allow us to fight pathogens and survive infectious diseases. Immune surveillance helps eliminate abnormal cells that might develop into cancer. Transplantation is a unique case where our immunity can work against the grafted organ or tissue. So, again, in the case that someone may have a blood or marrow cancer, leukemia or lymphoma, so the cells coming from their bone marrow are diseased. The treatment is to wipe out their bone marrow and replace it with someone else's. So, with that, you wonder about destroying someone's immune system, trying to reconstitute someone's immune system. You're trying to get a good matching so that you will have engraftment. Um, you may deal with issues of graft versus host disease, where the immune system can identify the host as foreign. You may have rejection where the host identifies the transplant as foreign. You can sometimes even get a graft versus leukemia effect where the new bone marrow fights the cancer as well. Again, when we deal with solid organ transplantation, we may be very concerned about anti-HLA antibodies, where HLA has become an antigen that someone has already responded to. So there are preformed um, antibodies which might cause a hyperacute rejection, or antibodies that might develop over time for chronic rejection, T cell mediated rejection, antibody mediated rejection. Um, so as we're developing additional testing, moving on from C4D staining to new markers and new post-transplant testing to make sure that people don't reject their organs. That's obviously what much of our lab work is associated with. So we did want to cover as much as we could today, just briefly, the portions of our immune systems that are relevant to our study of HLA.